Друзья, я думаю, что мы начнем. Мы еще пытаемся решить проблему с сидячими местами, но будем делать ее фоново, наверное. Здравствуйте, меня зовут Михаил, я куратор программы «Открытый университет». Сегодня я модерирую эту секцию. Здравствуйте. Сейчас у нас будет секция Лаури Алтанина, надеюсь, я правильно произнес имя, профессора Хельсинского университета, который расскажет нам о том, как генетические технологии меняют лечение, профилактику и диагностику рака. И я думаю, что мы можем его пригласить уже.
Thanks very much for the invitation. I really appreciate to have the opportunity to present a little bit our own work here, but also to tell you something about disease genetics, genetics in general, and perhaps a little bit something about the future regarding the use of genome, including perhaps some risks that may be associated with the use of genome information. So my title, From Genes to Genomes, Progress in Research on Hereditary Cancer, really focuses on the work of my research group, which is at the University of Helsinki, Department of Medical Genetics. However, I tell you a little bit about background of genetics because I do understand that most of you don't have a background in medicine. So we talk about genes and we talk about genome. Genes are like the one basic structure of the genome. A gene is usually a thing which you need to produce a protein. So first, this segment of DNA called a gene is transcribed into RNA and then translated into an amino acid chain, and after some folding changes you have the functional protein. And everybody has these about 20,000 genes, so we all share the same genes. It was a lot of work, though, to figure out what all these genes are. And this was done in a project called the Human Genome Project, which took like 13 years from 1990, and it was declared ready 2003. This was a project which was coordinated by the United States Department of Energy and the National Institutes of Health. And in case you wonder why Department of Energy wanted to fund a huge effort like this, it actually goes down even to the atom bomb and nuclear power and these sort of things. And because of that, Heritage Department of Energy had this task to do research into genes and in particular effects of radiation, DNA repair, these sort of things. And that is why they contributed quite significantly to this effort. And they had quite ambitious goals. It was planned to last 15 years, but as you can imagine, the technologies usually develop more rapidly than one would ever think. And they finalized the project a little bit before the schedule, so 2003, so 13, 14 years. Actually, it was a pretty accurate estimate, the 15 years. The goal was to identify all the genes. It turned out to be then 25,000. Originally, it was believed that humans would have much more genes, 40,000, perhaps 50,000. So the early guesses came down along with the progress of the project. So obviously determine the sequence of the 3 billion chemical base pairs that actually make up the DNA, so the very basic pieces of DNA. And importantly, store this information in databases so that researchers such as me and everybody could utilize that information for their research. So that's of course a very valuable aim, so that we have a tremendous impact from this project. Improved tools for data analysis is really important, and many of you, I guess, must be in that sort of a field. And for example, for my research, it's really, really important to have good collaboration with computer scientists, for example, because nowadays the amount of data is really massive. I'm a simple medical doctor, I don't understand what it means. but I need these people, such as those into computer sciences and bioinformatics, to explain. And indeed, one of the very aims of the Human Genome Project was to start developing tools for meaningful analysis. Also some other aims, including the ethical issues dealing with the process. The first draft sequence was published, I think, 2001 already in Nature. So in principle, we all have the same genome. So why do we have different phenotypes? Phenotypes being that somebody is tall and somebody is short and somebody has some sort of a disease and another person has not. Obviously, environmental factors play a role, but there's a lot of variation in the genome. So even though we basically share all the same genes, we have a lot of variation, so we are all, in a sense, very similar, and we are all unique. Except identical twins, they have their basic genome essentially as a replicate. And the variation really is very interesting when we think about different human phenotypes, because that's how it arises. And if we think about progress in hereditary cancer research or whatever genetics research, really the ability to detect the variation has been really a clue to success here. And for example, in 1998, only 4,000 common genetic variants were known, and the genotyping, so when you wanted to analyze these variants, you needed to analyze one at a time. And that was a lot of work. It was doable, but it wasn't like high throughput, was it? And in 2001, when the first draft sequence of the human genome showed up, one could see that there were about 1.4 million such variants, but still the methods to examine these were not very well developed at all. So you could have a look at perhaps tens at one time. And then in 2007, the DNA chip technologies made a breakthrough, commercial breakthrough, so they became sort of affordable for research, for example. And these DNA chips, they can measure about 1 million, 2.5 million variants at the same time with one experiment. So this was a big technological breakthrough, the DNA chip technology. So with that technology, you could measure essentially all the common variation in humans. What I have, what you have, and get a very accurate readout of all that. There is more variation than this, so there are more than 10 million variants in the human genome so far characterized of the about 3,000 million bases that the genome contains. As I said, the variation is then related to human diseases and traits. So this is how genetic diseases arise, because of the variation. Some of us have a better luck than others regarding this. Often when we talk about genetic diseases, we talk about these rare so-called Mendelian diseases. Mendelian means that it's either like dominantly inherited, so you have a 50-50 chance of being affected if your parent has the gene and there's a high penetrance. And there are these syndromes like cystic fibrosis, which is a recessive disorder, so you have to have 2 deficient copies of a gene to be affected. This child here having that disease, it's a very serious lung condition, and already young children are affected. And many others, Huntington disease, for example, one famous example of a degenerative neurological disease, but also cancer syndromes. There are many cancer syndromes which are sort of Mendelian, so that the cancer really runs in the pedigree in an autosomal dominant way. But also genes have a big effect, of course, on these common phenotypes, such as height or weight, these sort of things, intelligence probably, also cancer, common diseases, heart disease, schizophrenia, Alzheimer's, whatever. So one could say just about all the diseases, they have some sort of a link to genetics. All the traits have some sort of a link to genetics. So how to identify then the disease genes? Obviously, when all these efforts started, the tools were very, very simple. So as you can recall, we could measure just like one variant per time in one experiment, and it takes a lot of time to browse through the whole genome. So one started with these very simple phenotypes, right? You had a big family, you had a clear disease running in the family, a disease which preferably you could separate from non-genetic disease, such as that you have like a condition called familial polyposis, which is a colon cancer syndrome, and there the affected individuals, they have hundreds or thousands of little tumors in their colon. And that doesn't occur by chance. And if you have a family like that, you know exactly who is having the gene defect and who is not when they are about 30 years of age. So the polyps, it takes time to develop, so they are not there at birth. So utilizing these sort of families, big families, a lot of information, very simple, straightforward disease phenotype, one could already identify the first disease genes like almost 30 years ago. And we have done in Helsinki, for example, a lot of work like this. So here you, for example, see a pedigree. So this is the father, this is the mother who is affected. Then there is a bunch of children, some of which are affected, some of which are not affected. And then there is a simple genetic marker, one at a time, as you recall. And with this marker, the researchers get lucky and see that with this A allele, you see that the A allele segregates, it goes with the disease, whereas the unaffected individuals do not have it. So you can sort of conclude that, okay, the gene defect probably is going to be near this marker. It took a long time, even with these simple, straightforward diseases, but it was successful because even those early tools to genotype people provided enough power in these simple phenotypes. And by simple phenotypes, I mean these very straightforward, clearly hereditary diseases. And once you can map the disease gene, then you can do all sorts of stuff to actually isolate the gene and identify the mutation, the gene defect that causes the genetic defect, and that is still a lot of work. And this was all a very laborious process back then in 25, 30 years ago. However, this was a very successful effort, and also there some technological advances played a very big role. And a large number of these Mendelian disease genes, these traditional disease genes, have been mapped based on these approaches. However, if we think about common diseases which are, well, common in the population, heart diseases, whatever, which I just listed to you a little bit, for those, this family-based approach is not good at all. So if you have a family with ordinary heart disease, you cannot easily find a marker which would perfectly segregate with the disease because there are so many factors affecting heart disease. So simply this approach which I just showed you, it doesn't work because you will have in the pedigree individuals who actually do not have the risk allele, and your analysis is essentially powerless. So with the family-based thing, one marker at the time thing, you really cannot identify the gene defects for common diseases. You need some technological advances again to achieve that. And that achievement was the DNA chip that I showed you in a previous slide. So how this works then is that you have a large number of affected individuals, and affected, I mean 2,000 individuals with diabetes, or 2,000 individuals with colon cancer, or 2,000 individuals with schizophrenia, whatever is your phenotype that you are interested in. And then you have a good number of controls, perhaps 4,000 controls or whatever, and they should be population matched, of course. And you will see that when you genotype millions of SNPs, millions of these variants throughout the genome, so you do a genome-wide association study, as it's called, you will see that at some loci, your diseased individuals have more signal than the control population. There is some signal in the control population as well, but with appropriate statistics and appropriate validation sets and big enough data sets and enough informativity in your statistical analysis, that is, you can get a robust readout that here with the red thing in this chromosome, there is a locus predisposing to diabetes or cancer or whatever is your phenotype of interest. But this really required the discovery or the development of these DNA chips, which are able to measure the variants just like that in millions rather than tens or hundreds. So these are like common variants which are in the population. We all have them. We all have some risk alleles for whatever diseases. And if we think about rare variants, you cannot tag rare predisposing variants with this sort of an approach. For that, you would need a technology to sequence the whole genome from a huge sample collection similar to here. But you would need to sequence, you would need to get a readout of all the bases of the genome, because these rare disease-causing variants are not included, obviously, in these pre-manufactured SNP chips, which measure the known common polymorphisms in the population. So these would need to be sequencing-based. And we are soon there, and we somewhat are there already, depending, of course, on how much money one has to sacrifice to the analysis. But this here, this upper part, was a big advance, and that has been very cost-effective for about 10 years. And you see here some results of that, just some arbitrary. It stops to 2007 because, you know, it gets too overwhelming, but you can see how the technological advance, that about in 2006, these SNP chips, these DNA chips, were introduced to the research community. It really caused a flood of identified disease loci in just about any possible disease or common phenotype in humans that you could imagine, starting from cholesterol or obesity or Crohn's disease, which is an inflammatory bowel disease, diabetes, multiple sclerosis, you name it, it's there. Also height and these sort of things, not really diseases, but human phenotypes. There are plenty of loci, and these are very robust findings. So statistically reproducible findings. Not, of course, every single one, but in general, this has been very successful. What perhaps has been the problem is that you get many signals which are robust, but you get sort of an idea that, okay, the predisposition locus is here, but what really is the functional defect or functional effect which causes the disease predisposition? That is much harder work. We have done a little bit of that as well in colon cancer, but it requires like building an animal model and stuff like that. And you can't build an animal model very easily for all these phenotypes. So it's going to be slow to get there. But it will tell about the functional basis of the predisposition, which again provides you perhaps with some tools for developing targeted therapies. So if you understand how this disease arises in the population, if it's diabetes or cancer or whatever, you are of course better positioned to figure out how to treat it or even how to prevent it. And risk predictions are of course perhaps relevant at least at some point, in particular if you have some way of prevention. I need to monitor the time a little bit. I can see the clock here. So, the actual subject of mine is cancer, like I mentioned, and cancer is a disease of the genome, right? And it's not just the genome. There is a big environmental influence also. So if you smoke, that's very bad for you, right? That's why none of us do smoke. And the spectrum of cancers you get by smoking is really, really impressive. Other environmental factors, plenty of them. If you like to burn your food before eating it, that's one factor, certainly. Different viral infections is a very big factor. So all these environmental cues, they play together with your genome, And also there is a stochastic component, so if you have luck or not. And then there are certain processes where these genetic changes resulting from this environmental induced damage accumulate. I won't go into those in detail. But the idea is that here you have your germline genome, there you have the environmental load, you also have the mutation load which comes from cell division. So when you get older, your cells divide. When your cells divide, the DNA duplicates. When the DNA duplicates, errors are incorporated into the system because the duplication system is not perfect. So with each cell division, errors are introduced to the DNA even without smoking, even without getting bad viruses into you. So, so there is really nothing you can do about some of that mutation load. But there is, of course, a lot that you can do to some bits and pieces here. But the idea is that you have your germline genome, then mutations accumulate with time when you grow older, and with bad luck, you get cancer. But you can, of course, do something about that, like I explained. However, this process usually takes time. Cancer is usually the disease of the old, right? If we think about colon cancer or breast cancer or these common human solid tumors, prostate cancer, usually the people who get affected are perhaps, 60 years old, 70 years old, 80 years old. Of course, also younger people, but in principle, cancer is a disease of the old. If you think about cancer, the whole picture. There are many exceptions to this rule, of course, but this is the big picture. And the simple explanation to that is what I just explained. It takes time for the mutations to accumulate. It takes time for a neoplastic clone, a cancer cell to be produced. And thus, when you get cancer, you usually are old. So this is the age curve. So it starts rising reasonably sharply thereafter, about 50 years of age. However, we are talking also about hereditary cancer syndromes in this talk. So what is the difference there then? How is hereditary cancer different? Hereditary cancer is different because when a person is born with a hereditary cancer predisposition mutation, there is already one mutation in every single cell of that person, sort of waiting there. And that tremendously shortens the amount of time needed to actually produce the unfortunate outcome. So if this is the usual situation that, for example, there is a person without particular predisposition, he or she has two important genes which control cancer, which are functional, then with time in one cell, one copy gets inactivated. Still, you would need in that same cell another hit to get two copies of this cancer break inactivated, and then you get into trouble. Whereas a person who has a mutation already in every single cell, you can understand that such a person gets the cancer on average on much younger age. And thus, hereditary cancer is typically a disease of much younger generation, let's say in breast cancer, colon cancer, 40 years old, something like that. These cancer syndromes, they are reasonably rare in the population, high penetrance. You see these pedigrees with cancer, whereas the variants in the population predisposing to common cancers, these variants are of small effect. There are many, but very small effects, and these are very common. So we all have some. So this is what a high penetrance cancer pedigree might look like. This is an actual pedigree from Canada. You cannot see probably any details, but sort of the clinical features are obvious. Low age at onset, cancer in the family, multiple primary tumors more often than not. So for example, in breast cancer, if there is bilateral breast cancer, that should ring a bell. So it's not usual to have a separate cancer in both breasts. So it makes sense that if you have predisposition, these are the features. However, it is often difficult from one individual to say whether he or she has something like that. You would need a thorough family history. If the family is small, you don't have a family history at all, perhaps. So it's quite tricky, actually, to diagnose in clinical practice. We have had a look at the hereditary colon cancer in our colon cancer material, so unselected population-based material, so 1,000 patients. And we could identify a hereditary cancer mutation in 40 of these individuals, so 4%. Colon cancer is one of the common cancers. So 4% of them, it's not like very much, but it's significant for medical doctors. So certainly if there is a family history positive for colon cancer, the doctors nowadays, they should be alert, and genetic testing for these syndromes is now routine and should be performed when there is a strong suspicion, because then you can identify the at-risk individuals. Also, the treatment options are often different in hereditary cancer versus sporadic. This I actually already went through. So this susceptibility runs from very strong, like here, this is familial adenomatous polyposis, so you cannot perhaps see in detail, but this is carpeted by small tumors. So there is no normal bowel here at all, and it's carpeted by small tumors. There are probably more than 1,000 in this one specimen. So to this common predisposition where most people don't get cancer, but some people do, and it's sort of a polygenic thing. Like I explained, it has been for some time possible to actually identify genes for this population-based cancer predisposition also. So having a look at, let's say, 2,000 cancer cases, 2,000 controls, looking at these results from your DNA chip, you can see that, okay, some loci, some variants are more common in your cases than in controls. And when you have sufficient statistical evidence, your predisposition gene is there. And more than 20 of these low penetrance loci have been identified. We have done work mostly on this, which is near a oncogene called MYC. And I again look at the time. You can see here one interesting feature, and that's the allele frequency. It's 0.51, which translates into that 51% of our chromosomes carry that risk allele. So half of the chromosomes in this room, they have the risk allele. And because we all have two chromosomes, chromosome number 8, one from the father, one from the mother, about 3 out of 4 of us carry either 2 risk alleles or at least 1 risk allele. So these are really, really common. So we all have some of these risk alleles. And here is and the risk is small. So having an allele is about 1.2 times risk versus not having an allele. So that's not very much, but considering how frequent the change is, it's a big impact if you think about population. Similar to height or weight, some people are tall, some people are short, some people have many risk alleles, some people have few risk alleles, some people in the population have a rather high risk of colon cancer, and others have a rather low risk of colon cancer, if we think about genetic risk. And it's a bell curve. It's like height or something like that. Cancer risk essentially is a bell curve. Of course, in principle, if you knew that you were here, you could perhaps do something about that, perhaps avoid to burn your food and do stuff like that, perhaps go to a colonoscopy when you get 50 years old or something like that. These are not used widely at all, but in principle could be used for targeted prevention efforts. So now we can move a little bit to the sequencing-based approaches. And here really, since completion of the Human Genome Project, which was really an expensive effort, sequencing has experienced rather a dramatic drop in price. So about 2 million-fold decrease in cost over a decade. And about in 2005, 2006, it was discovered a new method or new approach to do sequencing. At that time, we had a reference genome, so we knew what a human genome is supposed to look like. So at this point, technologies were developed where you can actually randomly sequence little bits and pieces of a human genome, and then by bioinformatics tools, assembly it back to the reference genome and see what was in the patient or in the control person, whatever. And by utilizing these new approaches, sequencing became much less expensive. So that nowadays you can even commercially have your whole genome sequenced with perhaps about $6,000 or something like that. And of course the real cost is much smaller than that. And scientists have utilized this in cancer, in particular by sequencing cancer genomes. So cancer is a defect of genes, and we would like to get a comprehensive catalog of the genetic changes to understand why cancer arises and perhaps to find treatments and stuff like that. So all the major cancer types have now been sequenced, and more different subtypes are being sequenced, and this has really been a marvelous effort and produced a lot of useful data, some of which already has been translated. So from human genome to genome of the breast cancer, genome of colon cancer, and essentially every single cancer is unique. So that's a bit of a bad news, of course. So in every single cancer case, you have the genome of the patient, so the normal genome, which plays a role, and you have the genome of the tumor, which is unique for that patient alone, because only he or she has a certain composure of mutations within his cancer. So that makes disease or developing targeted treatments somewhat, of course, challenging. I won't go into details, but many of these driver genes driving cancer have been characterized, all different cancer types sequenced. We have a good view of the numbers of mutations and what kind of mutations they are. We can also get an idea from the tumors what has caused that particular cancer. Has the person been smoking? Is it a melanoma based on extensive sunbathing? You can see these signatures in the tumors. So that has been quite interesting. So indeed, like I said, we now have a reasonable catalog of cancer predisposing variants. So we know what are the variants in human genome which cause cancer predisposition. We also know a lot about the somatic mutations which cause cancer. And all this is potentially useful, and to the degree that some companies have started to offer like whole genome sequencing, for example, for people who are just interested in their health. They have some medically relevant problem which they would like to tackle, and in particular cancer patient sequencing, where they sequence the tumor and they sequence the respective normal tissue to understand what might have caused the tumor and is there any way of actually perhaps curing that. So what we can do with this information in cancer research in particular, the targets that have been identified mostly by sequencing are manifold. Last year in August, about 62 FDA-approved targeted therapies in the US. So these are getting to the clinic, but of course it is not unusual that they serve a reasonably small patient group. But in many cases, it has made a significant difference, and certainly one reason why the results in cancer treatments are improving is really based on these new targeted therapies. Could I actually ask from the organizers at this point, because we started the session a little bit late, so how much time would I actually have so I can finalize the talk in peace? 5 minutes still, okay. So I'll rush through the end slides then. Immunotherapies I should definitely mention. So in particular in tumors where you see a high mutation load, they are good targets for the immune system, but they usually have developed a way to escape that. And tackling that way has produced really nice results in multiple ways. So immunotherapy didn't really have a good name 10 years ago, 20 years ago, but now it really is one of the most promising ways of tackling cancer, and there are many detailed approaches which you can utilize to that end, so utilizing the patient's own immune system to actually fight the cancer much, much better. The germline predisposition is valuable to know. I skipped this slide in the interest of time. It is just about acceptance of genetic testing. In Finland, we have studied that, and we have noticed that in our high penetrance cancer families, people usually are pretty okay with the situation. If they hear that they have the risk allele and a high risk of cancer, they go to the follow-ups, and they don't seem to have like major psychosocial problems, so we have looked into that. But importantly, we also can offer these individuals effective cancer screening. So we have a national colon cancer screening program for these families, and it's rather effective. So we can reduce colon cancer, and we can reduce colon cancer-related mortality. We can almost get rid of it. And we also have an impact for overall survival. So Finland was the first country in the world to actually show it. Also, some chemotherapeutic approaches, such as aspirin, they might be effective in these cases also. Genome editing is now emerging as a possibility. So those of you who are interested in biotechnology, you would like to look into this. So nowadays it is getting more and more accurate, so we can actually replace even one base in the genome with another base. And this is basically a promising approach for many purposes, even for germline editing, though that would be, of course, ethically quite problematic. Indeed, that brings us to the last section of the topic, so to speak, which is like the issues that are there. Informed consent, this is our own informed consent form, which I have shared with many researchers in various languages because it has been a pioneering one. Essentially, we say in this that we may sequence your whole genome. We look into the cancer-associated items. We don't look at anything else specifically, but if we see something which is really important for you, would you like to know? And the principal lesson from this is that whatever you search from the DNA, think about it beforehand before you do the experiment, before you do the analysis. Think what the consequences are regarding the result, and make analysis only if it's well justified. Also, some people are excited about sharing their genetic information in various ways. I look at it very conservatively. I haven't sequenced my genome, but if I had, I wouldn't post it to Facebook. Some people actually are quite into these things, but at least it would be important for them to know that such information can have some unpredicted consequences. Very briefly, this was a science paper where the researchers actually identified some individuals which were in a population control database, anonymous individuals, but they had the DNA markers. They went to these databases publicly available for genealogy, and they were actually able to identify who these people are. Of course, they didn't publish the names in the journal, but they published their approach, and this was a bit of a shock. People who sequenced the famous cancer cell line, which was called HeLa, didn't realize that it is somebody's cancer cell line. It's actually Henrietta Last's cell line, and her family wasn't very happy about this. And there was a big row, and which was settled a few years ago about this. So the researchers just didn't come to think that when they publish the results of the cancer cell line, they actually publish someone's genome. And of course, such a publication affects also the close relatives. This is an example of a guy whose father was a sperm donor, and he was interested in knowing about his ancestry. So he used one of these ancestry services, and his Y chromosome, which runs with the male sex, was genotyped, and he could see from the databases, from this social media databases, that people with a family name of this have a similar Y chromosome. So his father might have had this family name. He had some information from her mother about the origins of his father. And combining these two pieces of information, he was actually able to identify the sperm donor who himself hadn't used any of these DNA genotyping facilities, but some from the same family had. And because the Y haplotype, the Y genotype, tags the family name, because both run with the male sex in the family. So this was quite unexpected again. And again, something that you wouldn't really think about it. Organ donors, well, if you donate your bone marrow, you also donate your genome in a sense. You can get, from your DNA, you can get the information, for example, from your looks. So this is, of course, forensic science, but also some artist in New York collected these used cigarettes and made sculptures of the individuals who had been smoking them, regarding or based on the genetic information that was available in there. So people are careless. They have to be careless for their DNA in this regard, but at least I would suggest that you wouldn't post it to social media. Also, things can get a bit silly even because people really don't understand how easy it is potentially to produce this sort of genome data, even whole genome based. So this is from a news flash, from a Finnish news flash. There is a forestry fair, and there is this politician who was in the fair, and they had a competition of chopping log, and he hit his arm, and there was some blood on the log, and because he was a famous guy, then the organizers felt that, okay, let's make this a bit of a nice occasion, and they actually auctioned this log there, and it costed 20 euros. And nowadays, this guy is the president of Finland, and they essentially auctioned the president of Finland's genome. So just some common sense. I mean, those of us who are not presidents, we don't perhaps need to care, other than let's not share our genomes without thinking first. But certainly those who are presidents, I'm sure, nowadays, at least I hope that they a little bit think about also these items, so that they wouldn't actually auction their genomes, or at least that it would cost more than $20. So thanks very much for your attention, and I'll be happy to take questions.
Вопросы? Можно на русском. Пожалуйста. Да, микрофон.
Hello. Mutation in DNA is just a part of the problem. Do you test in the RNA? Do you test in gene expression as well? Do you have in biobanking to test it further? Thank you.
This is, of course, a very good question. So in particular, if we think about characterizing the cancer phenotype, it is very important also to have a look at these additional layers of data. Genetic mutations is one part. One very big part is like epigenetic changes, such as, for example, methylation. So DNA is not only mutated in cancers, but it's in many times aberrantly methylated. And it means that if it's aberrantly methylated on certain sites, the regulation is different. So it changes the regulation. So in many cases, cancer is about aberrant expression of genes. And if there's, like, for example, methylation somewhere where it should not be, a gene is maybe silenced. And this is a very common way of getting important genetic or important changes towards tumorigenesis. Also, there are these other subsequent steps, such as how the proteins are degraded, for example. So even if everything else is reasonably fine, if there is some problem with protein degradation, the concentrations of the proteins are not going to be right. So it is a very complex problem. And to characterize the tumors, to understand them better, we certainly need all kinds of layers of data. You are absolutely right.
Еще вопросы? Окей. Спасибо, Лауре. Лауре, надеюсь, останется с нами в фойе, поэтому если кто-то не успел задать вопрос или не захотел задать его лично, публично, может задать ему лично. Спасибо большое. небольшое объявление. 10 минут перерыв. Через 10 минут начинается новая лекция.